Hello and welcome. Let's talk today about proteins. Let's talk about proteins. Now, when we talk proteins, I would like you to know, so when we talk proteins, proteins make up about 10 to 30% of a cell's mass and about 50% of the organic material in the body. So it's basically the structural material of the body. However, not all proteins are construction material. We'll see other functions that proteins are going to be involved with in relation to the body as we progress. Now, when we talk proteins, I'd like you to know they're also going to contain carbon, hydrogen, oxygen, okay, that we have seen before, nitrogen, we're going to see as well, and other elements exist also, such as sulfur and phosphorus, in relation to our protein structure. Now, our proteins are polymers, like we have described before, the term polymer. So they're going to be made up of a whole bunch of monomers. The monomers are going to be the amino acids, right? We've learned amino acids are the building blocks of proteins. So that's exactly what we're going to see. Now, when we go through and we talk about proteins, I want you to know that they also play a role in cell function. They also play a role in cell function. Proteins are going to include enzymes. They include enzymes which function as biological catalysts, which help to speed up chemical reactions that usually would take longer to occur, right? That's what enzymes do. Now, proteins are also going to be including hemoglobin in our blood, right? It's responsible for transporting gases, oxygen mainly, and the contractile proteins of muscle. You can see varied functions, right? Keratin makes up hair and nails. Collagen is going to strengthen our tendons and our ligaments. So when we talk proteins, they have the most varied function. You can see they have the most varied function of any molecule in the body, compared to any molecule in the body. Now, when we go through and we talk monomers are going to be amino acids. The bonds between the amino acids are going to be peptide bonds. So when we go through and we talk amino acids are the subunit monomers you can think of proteins. They are the subunit monomers of proteins, or you can define amino acids as the building blocks of proteins. There are 20 common types, 22 total, 20 common types. When we talk about the structure of amino acids, all amino acids except for one, which is proline, are going to have two important functional groups in their structure. So when we talk about their structure, you're going to see their amino acids are going to be joined by covalent bonds that are called peptide bonds. And they're going to contain in their structure, we'll see a basic group. The basic group is also called an amine group. So they're going to contain an amine group. And they will have an organic acid group. The amine group we'll see is going to be NH2. The organic acid group is going to be COOH. So that's why I told you guys to remember those functional groups that we saw before. Now, when we go through and we talk about amino acids, I'd like you to know an amino acid can therefore act either as a base, which is going to be a proton acceptor, or it can act as an acid, a proton donor. All amino acids are identical except for a single group of atoms called the R group. So they're going to have an amine group, an acid group. That's allowing them all to be similar. So they're all identical except for the R group. It's going to be, we'll see, the part in green. So here you can see you've got a central carbon to which is attached an amine group, and also on the other side an acid group. And then here you can see an R group attached to it as well. So Everything is going to be exactly the same in all the amino acids except for the R group. That R group changes, and as it changes, that's what then allows us to have different amino acids. So you can see here glycine, the simplest amino acid, for example, is only got a hydrogen. Now here, aspartic acid, instead of a hydrogen, it's got, you can see CH2 and then a COOH attached to it. And then here, lysine, even more different. And then cysteine, you can see there has sulfur. So they're all going to have an amine group, as we see in all the pictures. It's similar. And then a carboxyl group here we can see. And then an acid group here we can see. COOH, an acid group. And then the varying R group. So if you can understand that, it's easier to understand the structure of amino acids. So all amino acids are identical except for a single group of atoms called the R group. It's the difference in the R group that makes each amino acid chemically unique. Proteins are long chains of amino acids joined together. The amine end of one amino acid links to the acid end of the next. You can see here in this picture. The amine end of one amino acid links to the acid end of the next. Peptide bonds are the bonds between amino acids when they link together. So these are a type of covalent bond, we said, this peptide bond. So we have seen the type of synthesis reaction that's going to help to join them together before as well, the dehydration synthesis reaction, which joins two amino acids and forms a dipeptide, and you have water being produced. So if two amino acids are joined together, dipeptides are formed. 3 amino acids, tripeptides. Then when we have 10 or more, we have a polypeptide. Polypeptides with more than 50 amino acids are called proteins. Most proteins are also called macromolecules. Large, complex molecules which contain around 100 to over 10,000 amino acids. And then the term glycoproteins or glycolipids, they're going to be basically, glycoprotein is going to be a protein that has a glucose attached to it. Glycolipid is going to be a lipid that has a glucose attached to it. So then the opposite of putting these two amino acids together, we have our hydrolysis reaction, which will then help to break down the dipeptide or tripeptide or polypeptides to their constituent building blocks, the amino acids. Now let's look at the structural levels of proteins. There's 4 structural levels that I would like you to know about. So here, if we were to get basically amino acids and start putting amino acids together, okay, we're going to form a primary structure. A primary structure in the structures of protein hierarchy. A primary structure is a polypeptide chain which is found as a linear sequence of amino acids, and it resembles a strand of amino acid beads, you can kind of think of. It resembles a strand of amino acid beads, and it is the backbone of our protein molecules. Proteins aren't normally found as this structure. Instead, we add more and more amino acids to them, and then this will cause them to twist or bend upon themselves to help form then the more complex structures like our secondary structure. So our secondary structure is a more grown primary structure. So it's a primary structure which has had more amino acids added to it. It's more complex. It's twisted, you can see, or bent around itself. So the primary structure we start adding more and more amino acids to it, and we get then our secondary structures. Our secondary structures can be of 2 types. We can have number one, an alpha helix. An alpha helix is going to be the most common type of secondary structure, and it resembles a slinky toy. It resembles a slinky toy or the coils of a telephone cord. I don't know if you guys know what a telephone cord is these days, but kind of looks like that. It's formed by the coiling of the primary chain. And it's going to be stabilized. It's going to be stabilized by these hydrogen bonds you see in here. It's stabilized by these hydrogen bonds. So you can have either this type or you can have the second type, the beta pleated sheet. Now the beta pleated sheets, these are primary polypeptide chains that are not coiled. Instead, they are lined, or you can say they are linked side by side. They're linked side by hydrogen bonds, forming a pleated or ribbon-like structure that resembles what we can refer to as an accordion's bellows. It resembles an accordion's bellows. So if we keep adding on more and more amino acids, then what we're going to form is the tertiary structure. So when we talk tertiary structures are going to be formed again by adding more and more amino acids to our secondary structures. So they're formed when the alpha helical or beta pleated regions of the polypeptide chain are going to fold upon one another. They're going to produce a compact ball-like structure or a globular molecule, as we can see here in the picture. So again, add more and more amino acids, takes us to then the quaternary structure. When we talk quaternary structure, this is a complex protein. This is a complex protein. It's formed when you have more than one polypeptide chain. So basically when two or more polypeptide chains come together or aggregate. When two or more polypeptide chains aggregate or come together. So here you can see a quaternary structure. An example will be hemoglobin. Next thing I'd like to talk to you about protein denaturation. So here you can see primary structure, nice linear chain. And then we've got our secondary structure, our tertiary structure, and then here is our quaternary structure, So protein denaturation. I'd like you to know protein denaturation is a process in which hydrogen bonds are broken. Hydrogen bonds are broken by many chemical and physical factors. So it can happen by many different chemical or physical factors. And some of these chemical or physical factors can include factors such as excessive acidity or excessive temperature. And what this does is it's going to cause proteins to unfold. It'll cause them to unfold and lose their characteristic shapes. So rendering them then useless. Let's talk. So when we talk denaturation, globular proteins unfold and they lose their functional 3D shape. And then what happens is that active site gets destroyed. So they're not going to be functional. The active sites get destroyed. And this can be caused again by a decrease in pH or increased temperatures. This is usually reversible if normal conditions can be restored. Irreversible if the changes are extreme. It's like cooking an egg. You can't reverse that. You've had total protein denaturation take place there. Let's talk enzymes. Now, when we talk about enzymes and enzyme activity is what we're going to look at. Enzymes are proteins, as we've described. They're proteins or protein molecules that are going to act as biological catalysts, or we can refer to them as protein catalysts. Catalysts are substances that are going to regulate and accelerate. They will regulate and accelerate the rate of biochemical reactions, but they themselves are not used up in the reaction, nor are they going to be changed in the reaction. So they go in and they make their way out of the reaction without ever having been changed. but the reaction is going to have taken place. Enzymes, I'd like you to know, are going to end in the suffix ase. They end in the suffix ase. So one easy way to recognize, not every single one of them, but most of them, is you'll see they end in that ase. So here we can go through and we can talk more about enzymes. Enzymes are globular proteins that are going to act, we said, as biological catalysts. And here you can go through, and we'll go through all of these factors here as well. We'll check them out. I'd like you to know enzymes are going to contain two parts. They contain two parts. One is an apoenzyme. That's the protein portion. That is the protein portion. And a cofactor. Or coenzyme, we can say. And then the cofactor or coenzyme can be an ion or an organic molecule that's needed to assist in the reaction. Now, when we talk about enzyme action, we'll see chemical reactions are going to require energy. They're going to require energy, which we refer to as activation energy. Activation energy is the amount of energy that's needed to help break the bonds of the reactants so that way they can rearrange themselves and become the product. Kinetic energy. Kinetic energy is what pushes the reactants. One way to increase the kinetic energy is to increase the temperature. But higher temperatures are going to denature proteins, as we just talked about. This is why high fevers can be a serious event. But when we talk temperature changes, that's not really common in the body, because in the body the temperature is usually stable. Enzymes are going to allow reactions to occur at normal body temperatures by decreasing the amount of activation energy that's going to be required. Now, enzyme action is going to happen by 3 steps. It'll happen by 3 steps we'll see first. So step number 1, the substrates. Substrates are going to bind to the enzyme. So substrates are the substances on which an enzyme acts. So here you can see are the substrates. For example, these are 2 amino acids. And they're going to come and they're going to bind on the active sites of this enzyme. So substrates bind to the enzyme active site, forming an enzyme substrate complex. The enzyme then changes shape. It changes shape to hold the substrates in their proper position. Second, then, the enzyme substrate complex undergoes internal rearrangements that form the products. And then step 3, the enzyme releases the products of the reaction. The enzyme releases the products of the reaction. And then you can see here, the enzyme is going to leave the reaction just like it was when it went back. And here you can see the enzyme is going to leave the reaction just as it looks when it came in. So it itself doesn't get used up in the reaction or used. Here you can see it helped to create the peptide bond and form a dipeptide. So again, the enzyme, it's not changed. The enzyme is not changed, and it is available to catalyze another reaction right away. And it's available to catalyze another reaction. So you have the same thing right inside of here. It's basically a lock and key model we see here. And then here we have a induced fit model, basically depicting the same thing. The product is formed and the enzyme itself is not used up. All right, let's talk about the regulation of enzyme-mediated reactions. Now, the rate of an enzyme-mediated reaction in the body is going to depend on three major factors. These three factors include, number one, substrate concentration, as we can see here. Number two then is enzyme concentration. And number three then we have enzyme activity. So let's talk first substrate concentration. Substrate concentration is going to be, again, so these are, and we're talking enzyme-mediated reactions being regulated by three factors. So the first factor that can regulate an enzyme-mediated reaction is the concentration of the substrate concentration. Now, it can be altered by changes. So substrate concentration can be altered. Substrate concentration can be altered by changes in diet or the rate of absorption in the intestine. Intracellular substrate concentration can be altered by reactions that use or make the substrate. The rate of an enzyme-mediated reaction increases as substrate levels increase until it reaches a maximal rate, which then it remains constant despite the concentration increase. This level, when it gets reached, is called saturation. Next, then, we have enzyme concentration. When we talk enzyme concentration, at any substrate concentration, including saturation, the rate of an enzyme-mediated reaction can be increased by increasing enzyme concentrations. Number three is enzyme activity. A change in enzyme activity occurs when either allosteric modulators bind to a receptor to change its response to a stimuli. That happens in, for example, benzodiazepines. Example, Xanax. It makes nerves in the brain less sensitive to stimulation, thus the calming effect. Or covalent modulation, which occurs in which enzymes are regulated by transfer of molecules or atoms. Next, then, let's talk about nucleic acids, DNA and RNA. Now, nucleic acids, DNA, deoxyribonucleic acid, and ribonucleic acid, RNA, is what we're going to talk about. So DNA and RNA, they are polymers. The structural units, the monomers of our nucleic acids are called nucleotides. So our nucleotides come together to give rise to nucleic acids. They're the largest molecules in the body, and they're going to contain carbon, hydrogen, oxygen, and even nitrogen and phosphorus in their structures. Each nucleotide consists of three components. Number one, you have a nitrogen base. Number two, you have a pentose sugar. And number three, you have a phosphate group. So here we can see an example of a nucleotide. Here we can appreciate the phosphate. Here we can appreciate the sugar. And then here we can appreciate the base. So same thing here, base, sugar, and phosphate. Three components helping to construct a nucleotide. Many nucleotides come together to form then our nucleic acids. There are five major varieties. of nitrogen-containing bases. You have number one, adenine, abbreviated with a capital A. Number two, guanine, abbreviated with a capital G. And adenine and guanine fall under the category of purines. They are purines. Next, then we have cytosine, capital C, thymine, capital T, and we have uracil, capital U. Uracil we're going to talk about in relation to RNA. And uracil will take place of thymine. So in place of thymine, you will have uracil. in RNA. So there's no thymine in RNA. And there's no uracil you can see here in DNA. So that's why they're not mentioning it here. But I'm just telling you that there's 5 bases, so you can get them all in your mind right now. And now you know, okay, uracil is only going to be used with RNA. Adenine and guanine are purines. Cytosine and thymine and uracil will be considered pyrimidines. They will be considered pyrimidines. Adenine and guanine are 2-ring bases. They have 2 rings in their structure. So here you can see there's 2 rings, so this has to be either adenine or guanine. Here's only 1 ring, so it's got to be thymine, cytosine, or uracil. So again, adenine and guanine are 2-ring bases, and they are called purines. Whereas cytosine, thymine, and uracil, they are single ring structures, and they are called pyrimidines. DNA is a double-stranded structure. It's a double-stranded structure, or we can refer to it as a double chain of nucleotides. We can also refer to it as a double chain of nucleotides. The pentose sugar in DNA, the pentose sugar of DNA is deoxyribose. It is deoxyribose. The pentose sugar of ribose, the pentose sugar of RNA is going to be ribose. So the pentose sugar of DNA is deoxyribose, and the pentose sugar of RNA is going to be ribose. The 2 nucleotide chains are held together by hydrogen bonds. They're held together by hydrogen bonds you see right here in the middle. They're held by hydrogen bonds, and the hydrogen bonds are going to be found in between the bases. They're found in between the bases. So what this creates is a ladder-like molecule. You can see here it creates a ladder-like molecule, or a double helix, Now when you talk about base pair bonding, bonding of the bases is very specific. Not anything and everything is just going to go and bind to each other. You will see adenine always binds with thymine, and cytosine is always going to bind with guanine, or vice versa. Okay, AT, TA, or GC, CG, same thing. Now here you can see when you talk the bonds between adenine and thymine, there's 2 hydrogen bonds there. But the bonds between cytosine and guanine, there's 3 bonds there. So you need to remember there's 3 bonds between cytosine and guanine, where there's only 2 hydrogen bonds between then adenine and thymine. Okay. So next then, I want you to know DNA is going to provide instructions for protein synthesis. It's very important that you know that. DNA provides instructions for protein synthesis. So what is protein synthesis? The synthesis of proteins, making proteins. Who gives the instructions of what the proteins need to look like in our body? DNA does. The DNA is going to replicate before cell division. So before our cell divides, our DNA is going to replicate, ensuring genetic continuity, right? Making sure that the cells that are going to come afterwards have this genetic information in them. So again, here we can appreciate a DNA strand. RNA, when we talk RNA then is going to have 4 bases. So in place of thymine, here we have uracil. So AUCG when you talk binding. The pentose sugar, ribose sugar we said. RNA is a single-stranded molecule. It's mostly active outside the nucleus. We're going to look at that. There's actually 3 varieties of RNA that are going to carry out DNA's orders for protein synthesis. There's 3 different RNA molecules we're going to check out that are going to carry out all of the instructions DNA gives for making protein. And those three include messenger RNA, transfer RNA, and third, ribosomal RNA and ribosomal RNA. So jumping back here, you know now the differences between DNA and RNA. If I was to give you a DNA segment, and I told you this segment I'm giving to you, I want you to find the complementary segments to them. Let me just, okay, we'll do it right here. So if I was to give you this segment, A, T, G, A, and ask you to give me the complementary segments to this segment here. Give me the complementary chain to this segment here. So what would bind to the A, we said? T, very good. What's going to bind to the T? A. And that's if I'm asking for a complementary, right, on DNA. Let's see, we're going to change it up a bit. So here we would have C, and here we would have T. Okay, so let's keep the same segment there. So that's if I want a complementary DNA strand. Now let's say I want an mRNA. Let's say I want an mRNA strand complementary to this strand I've given you here. So what are you going to give me here? Since no T in RNA, so here we're going to have U. A, C, U. So you have to listen to see what I'm asking for, or whatever the question is asking. Is it going to tell you, oh, can you give me a complementary strand to this DNA strand? Can you give me a complementary DNA strand to this example DNA strand? Or can you give me a complementary RNA strand to this example strand? And that's where then you would have to remember, oh, in RNA, there's no T. And so make sure you fill it in with U's. Now, RNA is chiefly located outside the nucleus. It's single-stranded, we talked about. It carries out the orders for protein synthesis, 3 different types. All right, awesome. Let's move to ATP. Let's talk ATP. Now, when we talk ATP, ATP stands for adenosine triphosphate. A lot of acronyms like to use DNA, RNA, ATP. So you got to know what they're standing for. In your exams, when you write your essays, you can't just use abbreviations, because when you're writing your essays, you have to remember you're writing these essays to people who don't have any idea of what ATP is. So you can't just say, oh yeah, well, ATP, that would be unfair. Like if I was to use that with you guys, you would question the same thing. So here we can see ATP stands for adenosine triphosphate. So on your essays, you have to either make a little legend on the side and explain what it means, and then you could use your abbreviations, I have no problem, or you can explain it in the essay, just like you see here, you're talking ATP, and in parentheses you can put adenosine triphosphate, and then after that you're welcome to keep using your abbreviations, as long as you explain one time what those abbreviations mean. So ATP, I want you to know, stands for adenosine triphosphate. ATP is the primary energy transferring molecule in cells. And it provides a form of energy that is immediately usable by all body cells. The energy released during glucose catabolism is coupled to the synthesis of ATP. In other words, some of this energy is captured and stored as small packets of energy in between the bonds of ATP. So when those bonds break, energy gets released. ATP is made up of an adenine. It's made up of an adenine, which is going to be a base. So here you can see that. It's made up of a ribose sugar. We could see that. And 3 phosphate groups. Breaking the last phosphate group, breaking the last phosphate bond of ATP leaves you with ADP, adenosine diphosphate, two phosphates. And energy is going to be released. ADP can release some energy too. And if it does, it becomes AMP, adenosine monophosphate, because of one phosphate. Now, ATP supplies, there's a little bit of energy in here as well. It's not as much energy as we would have in here, but there is a little bit of energy there, so we can use that energy there also. We can't go any further down now. ATP supplies are replenished as glucose and other fuel molecules are broken down. The same amount of energy that is released when ATP is broken down must be captured and used to reverse the reaction to reattach phosphates to reform ADP, and then adding another phosphate to reform then ATP. ATP allows a lot of life's processes to occur. ATP allows a lot of life's processes to occur. For example, it makes and degrades molecules. It transports substances. It transports substances across a cell membrane. ATP allows a lot of life's processes to occur, like, or for example, it makes and degrades molecules, transports substances across cell membranes, and allows muscles to shorten. So it's very important. And there's another source of energy we have in the body. It's ATP's cousin, GTP, guanosine triphosphate. I want you to be familiar that there is another source. However, we're not going to go into it as much in detail as we did here with ATP. So here you can see ATP being used to power pumps, helping muscles to contract, and even allowing chemical reactions to occur.